So welcome everyone to this week's CCNB seminar meeting, where we are very happy to welcome Ryszard Auksztulewicz. I was just thinking about when it was then we met the first time, Ryszard, and it was like I was joining the lab as a research assistant while I was doing my master studies, and Ryszard was doing his PhD in the group, and we were like collecting data together. So it was like a long time ago. And this was at the time.
So yeah.
It was like at the times when Ryszard was part of the Berlin-minded brain school and did his PhD here in Berlin back in time with supervision of Felix Blankenburg, mostly where we were located at the Bernstein Center for Computational Neuroscience, and then it was associated also with the Charité mostly. Then afterwards, Ryszard moved forward, had like some stays abroad, but most importantly was then moving for a postdoc to the Wellcome Trust Center for Neuroimaging, UCL in London, to work with Carl Friston. And from there, moving forward to do another postdoc in Oxford, mostly working with Kia Nobre. And then from there, moving to Hong Kong, where he was for multiple years, and now we are very happy that he's back to Germany. Was joining the group of Lucia Meloni and is currently affiliated to the European Neuroscience Institute in Göttingen. And he has over all of the time been working in the framework of predictive coding and using advanced analysis and modeling techniques to be applied to empirical data in the context of perception and consciousness processes. And so I'm very much looking forward to your talk today and hear about your recent research and future ideas. So great to have you, Ryszard Auksztulewicz.
Great, thank you so much, Timo, and thanks everyone for joining. Let me just share the screen. So actually today I think I will present kind of a relatively well spread out overview of what I've been doing since moving out of Berlin. So I will actually start with a study that I did at Phil with Carl Friston, and we will take it from there. Just a quick remark, I'm actually recovering from COVID, so if I cough, please excuse me. Hope it'll be fine. So today, I will give a brief introduction to predictive processing, but actually since this is a part of a bigger talk series on active inference and predictive coding, I think I won't be spending too much time on that. And then I will split the talk into several different parts, moving from sort of very strictly working in the predictive coding framework, where we used some generative biophysical models to analyze MEEG data and to answer some more mechanistic questions about specific hypotheses regarding predictive coding. And then I will move a bit away from that and move into more sort of general predictive processing framework and how we can study that with different methods, but also in different species, for example. So this is what I've done quite recently in Hong Kong. And we will talk about how prediction is modulated by other cognitive factors, such as attention. I will also talk about how prediction can multiplex with memory representation. And finally, how different kinds of predictions can actually be implemented in the cortex. So I don't think I need to really introduce this very general topic of predictive coding, but just so that all of us are on the same page, I think most of us would agree that the very traditional bottom-up view of perception as indeed a bottom-up process where different features at different levels of abstractions are integrated from sort of lower level to higher level stages of processing. This is no longer really an accurate model. And so this model has been refined in the early 2000s by positing the importance of top-down loops or recurrent processing, where at each stage there is perhaps some additional modulatory influence of top-down feedback from higher order regions, and this feedback might nuance or refine representations at intermediate or lower levels. But predictive coding is really flipping this traditional model on its head, and it actually posits that most of the heavy lifting in perception is actually in top-down direction. So higher order regions are thought to send some predictions via descending connections to lower order regions. This is supposed to occur throughout the cortex, but also even at subcortical regions. And at each stage, they are met with the incoming inputs or with the incoming prediction errors. They are then compared with these inputs, and the resulting comparison is the prediction error, which is then sent back up the hierarchy. And this prediction error is used to refine the model or optimize the model, the generative model that the brain entails about its environment and about the causes of its sensations. So this predictive coding model has been formalized both mathematically, but also has been mapped onto some most likely canonical microcircuits. So basically the most likely cortical mechanisms of predictive coding. And so here, for example, I mean, here I'm just plotting a figure from a review paper, but there's also a really good paper by Andrzej Bastos in Neuron 2012 that describes these canonical microcircuits for predictive coding. So the idea here is that predictions and prediction errors can be actually mapped onto different units. They can be mapped onto mostly pyramidal cells with some important role also of inhibitory interneurons. But the key thing is that there's some asymmetry, for example, between prediction errors being signaled by mostly superficial layers. And so this is represented here, for example, where these superficial layers are then sending prediction errors up the hierarchy. And predictions, on the other hand, are thought to be signaled by deeper layers, which are then sent back down the hierarchy. And there's also another asymmetry in terms of frequency bands. So it's also thought that prediction errors are sent in higher frequency bands, for example, in the gamma frequency range, whereas predictions are mostly thought to be mediated by lower frequency bands, such as alpha and beta band. Is this working? Yeah. But as we all know, prediction is not the only sort of top-down mechanism in perception. And it can be compared and contrasted with other kinds of top-down influences, such as attention. So actually, so let's just start with definitions. Prediction is typically thought of or defined as utilizing some statistical regularities from the environment to guide perception and behavior. On the other hand, attention is mostly thought of as prioritizing contextually relevant information. So hopefully right now you are prioritizing this particular talk and not any of the other multiple stimulus streams that you might have around you. So actually, at the level of behavior, both prediction and attention have been thought to have similar behavioral effects. They both enhance detection of stimuli, they both facilitate recognition. But at the level of early neural evoked responses, they actually have opposing effects. So prediction is typically thought to attenuate the amplitude of evoked neural activity, while attention is thought to actually enhance it. So this discrepancy has been to some extent theoretically integrated in the predictive coding framework, where prediction, the effects of prediction on weaker early evoked responses were linked to a low discrepancy between the top-down inputs and the incoming prediction errors, while the effect of attention has actually been modeled with a different term. So this term is quite important for predictive coding scheme, and this is called the precision or so the inverse variance of prediction error. And so this precision term also linked to gain, such as neuromodulatory gain. This is thought to scale prediction error signaling and to influence the model, so optimize the model more if these prediction errors are precise, for example, attended. So what follows from this is that prediction errors actually should be scaled by attention. They should be modulated by attention. And this is an empirical hypothesis that at the time when we came up with this model was actually not very widely accepted. So the consensus in the field at the time was that prediction error responses or predictive coding is a relatively automatic process. It's a relatively low-level process that occurs even in states like coma under anesthesia, which I will also talk about later on. But the key thing was that it was supposed to be pre-attentive, so basically operating almost independent of attention. But so we aimed at testing this hypothesis. So we devised an empirical study, which we ran in humans using MEG. And in the experimental paradigm, we orthogonally manipulated attention and prediction. So prediction was here in the auditory modality. We implemented this as a roving oddball sequence of tones. We had basically over the course of many trials, we had tones repeating over and over again, and then being replaced by another tone with a different frequency. So that new replacement, a new tone with a new frequency, that constitutes a sensory deviant. And then as you repeat the same tone over and over again, it becomes expected, so it becomes a standard. So our manipulation of expectation and prediction was the difference between sensory deviants and standards, and it's quite typical for the literature. However, the way that we manipulated attention was perhaps slightly less typical. So here we actually implemented it as temporal attention, so attention to events in time rather than, for example, space or different features of stimuli. So the way we did it is that in each trial, there were 2 time windows that participants could attend to, an early time window and a late time window. And in each block, actually, we gave them a cue, basically which time window would be the one that they should report. So they only paid attention to tones occurring in one of these 2 time windows. And actually in each trial, we set the probability of each tone occurring in a particular time window at 0.5. So that meant that in a given trial, there could be either 1 tone in the early time window or in the late time window, or 2 tones or 0 tones. And the response prompt was just to press a button if there was no stimulus in their attended time window, and this was done to minimize some motor preparatory artifacts. So in terms of MEG responses, here I'm just plotting the main effects of both attention and expectation on the event-related field amplitude. So the plot here in the upper plots present the topography of the effects, and the lower plots present the time course, where the x-axis is also actually spatial, so left to right, but the y-axis is the time axis. And so as you can see here, actually both main effects were significant, but they were dissociable from each other. So the effect of expectation, the difference between deviance and standards, was very similar to a traditional sort of a mismatch response, topography and latency. But the effect of attention was actually much earlier, and so already the first peak of the evoked response was modulated by attention. But crucially, what we hypothesized was that if prediction errors are indeed scaled by attention, that they should interact with attention. So that's also what we found. So basically, looking at the interaction, we found that mismatch responses were in our paradigm only significant if these time windows were attended, but not when they were unattended. So to explain this finding, we turned to computational modeling. And so here we really wanted to test where in the network these effects might occur and how they can be then linked to our original hypothesis in terms of predictive coding. So to this end, we used dynamic causal modeling, which is a generative model. This should be here for neural responses, not for behavioral responses. This is an important point, that here we use a generative model, a computational model, to actually explain our data. In this case, our data are neural data, neural activity signals. So in terms of, or in case of MEEG, we can actually use the very high temporal resolution of the data to test quite detailed mechanistic hypotheses about sort of the network connectivity parameters that might have given rise to the effects that we see at the sensor level or the scalp level in case of EEG. So here, our data that we try to explain are time series and actually entire scalp or sensor level topographies of mismatch responses in the attended and in the unattended condition. And the kind of ingredients, so basically the states and parameters of the models that we use to fit to the data, to explain the data, are at several different scales, from the macro scale, which is basically the connectivity patterns between different brain regions, via this meso scale, which is under the assumption that each region is composed of several different neural populations. This amounts to, for example, connectivity between these neural populations and things like gain parameters. or how sensitive a particular population is to inputs and how much it will scale the outputs. And then there are also some parameters at the micro scale, for example, time constants and these kernels that describe sort of synaptic dynamics. All of them are under the assumption that we can measure some activity from large populations of neurons. So these are not spiking network models; these are more neural mass models in this case. And another, since we are now in this generative modeling framework, this is also actually a Bayesian framework. So typically we have several models. We have a model space of alternative models that we try to fit to the data, and then we basically compare them using some Bayesian model selection procedure. And we find either one single model or an average of the most plausible models that explain a lot of the data, but at the same time are as parsimonious as possible. So we penalize the models for complexity, but reward them for accuracy in explaining the data. So just as an example, we might fit several models to the data. For example, a null model, model 1, in which we don't have any free parameters in terms of connectivity between regions. So we actually shouldn't be able to fit the differences between, let's say, attended and unattended mismatch responses. We also have models in which we, for example, can assume that all differences are due to top-down connectivity, other models in which we vary both forward and backward connection strengths, and finally, we can also add some gain modulatory parameters to these models and compare them with the other models. So in this particular study, we found that the winning model was actually showed an interesting dissociation between attention and expectation. So we could link these two different effects to different model parameters. So attention was actually linked to a top-down effect as well to an disinhibitory effect at the early auditory cortical stages, so in primary auditory cortex. But on the other hand, expectation was linked to no such gain effect, but more to both bottom-up and top-down connection modulation between different regions. And this is just to show you how well these models can fit the data. So this is now showing both the actually measured signals in the top two panels and the signals predicted by the model in the bottom two panels, where each, well, the x-axis is the time axis and the y-axis is actually one MEG sensor in each row. And we have both attended and unattended conditions, and typically we can explain more than 90% of the variance in our data with these biophysically realistic models. So to conclude this first study, we could show that temporal attention modulates auditory mismatch responses. We actually then replicated the same finding using very similar methods also in somatosensation, actually in nociception. So we looked at mismatch responses to pain stimuli. And so the result so far is that actually expectation can be still thought of as a comparison between predictions and prediction errors because it's both top-down and bottom-up connections, but attention has been linked in this case indeed to increased sensory precision consistent with our hypothesis. So in the second study, we thought of extrapolating this finding more to a more dynamic context in which we're actually looking at contextual relevance of predictions. So imagine you're walking through the beautiful Dahlem to reach the university, and all of a sudden you hear a loud honk. So in some contexts, this means that there's a car probably traveling a bit too fast and it's honking at you, perhaps you're crossing the road. So in this case, it will actually be very relevant to have some spatial and temporal predictions about where you might be in relation to this car in a few seconds. But in other contexts, perhaps not as likely in Dahlem, but more in, for example, Oxford, where this study was conducted, a car honking might actually mean that it's stuck in traffic, and so your spatial temporal predictions about where you are in relation to the car in a few seconds, that's not as important, so they're not relevant in your context. In this study, we try to actually disentangle the effects of predictability of a stimulus and the relevance of these predictions in dynamic contexts, where both temporal and spatial predictions could change over time, and either the temporal predictions or the spatial predictions were relevant at different parts of the task. And we wanted to ask the question whether predictability and relevance constitute independent source of information, or if they are interactive, such that the relevance modulates the influence of predictability. And we also wanted to look at their neural correlates in terms of signaling predictions, prediction errors, or both. So basically, which of these correlates are modulated by relevance. So in this study, this was also an MEG study in healthy human volunteers. We had a slightly more complex paradigm. And so we basically had an auditory cue and a visual target in each trial. The cue had two features and the target had also two features. So the cue had, it was composed of a pair of tones. They could be either high or low in pitch. So that was one feature. And they could also have either an ascending or a descending pair. So this is what I will call composition. And the visual target, on the other hand, it was located either on the left or on the right. So that's the location feature. And it could also be presented either early or late relative to the cue. So that's the latency of the target. So first of all, we introduced some contingencies between the cues and the target. So the pitch of the cue could predict the location of the target, and the composition of the cue could predict the latency of the target. Now, to manipulate relevance, we actually blocked task instructions such that in some little blocks of trials, the spatial task was important. So basically, participants pressed a button to categorize the targets as appearing on the left or on the right, and in other blocks, the temporal information was important. So they were pressing a button to indicate if they saw the target early or late relative to the cue. And over the course of the experiment, the predictability levels, so the cue validity levels, could change unknown to the participant. So this was all done in an implicit context. So for example, just to walk you through an example experiment, we could start with, let's say, 20 trials in which high pitch would predict the target occurring on the left, for example. And let's say the ascending composition would predict with 70% accuracy that the target will appear early. And we are starting, for example, with a temporal task. And then, for example, after 40 trials or so, perhaps one of these contingencies, let's say the composition to latency, will switch from 70% to 30%. So now an ascending cue would predict a target occurring late rather than early. And then we move on and we, for example, switch the task. So now we give participants a different instruction. And then we move on, another contingency might change. So now pitch is completely unpredictable with respect to the target, and so on. So we basically have this implicit learning paradigm in which the predictability levels change, but the only thing that the participant actually knows is what to pay attention to. So just the contextual relevance is important. So first we looked at behavior, and we found already at the behavioral level that predictability effects were modulated by task relevance. So basically the spatial predictability level, strong meaning 90%, weak meaning 70%, and none meaning 50%, the effects were only visible in terms of accuracy when participants were performing the spatial task. And similarly, the effects of temporal predictability only came out when participants were performing the temporal task, but in the other task, in the irrelevant context, there was no significant effect of predictability. So now to explain these findings and then to link them to neural activity, we used a different kind of a generative model. So this time, instead of trying to explain neural data, we actually try to explain behavioral data. So the model here is called the hierarchical Gaussian filter. As data, it can take many different kinds of data, actually. So it can take typical behavioral data, for example, such as reaction times, but also other kinds of behavioral data, like error estimates. It can also take actually physiological measurements, for example, skin conductance response or pupil size, or any other things that you can actually measure on a trial by trial level, and that you can somehow link to an underlying belief updating process. And so this belief updating process is what actually constitutes the model. So the hierarchical Gaussian filter is another Bayesian model, but this time of beliefs rather than of neural activity. And the hidden states and parameters of the models are basically some beliefs or predictions at different hierarchical levels in the of the task. And the parameters are learning rates. So basically how these predictions are updated over time. So in our case, the model had three possible levels. At the lowest level, the beliefs or the predictions that a participant could make on a given trial was about where or when the target will occur in that trial. At a slightly higher level of contingency, the participant could form prediction about what is the current Q validity level. So for example, is my Q predicting the spatial location of the target with a very high validity or with a very low validity, or is it completely unpredictable? And the highest level, perhaps you have noticed that actually we also manipulated that in the trial. So there were some in the experiment rather. So there were some portions of the experiment where these Q validity levels changed quite slowly, and others where they changed quite fast. So this is called volatility. And so at the highest level in our model, we also allowed our model to track some beliefs on how fast the environment is changing. So the volatility of the environment. And so the model is then linked to a so-called response model, which basically then translates directly to predictions of behavior in our case. So in our case, the response model was just sigmoid translating these predictions about the most likely target directly to the button press, basically to a trial by trial button press, so task performance. So the model space was also, just as in the DCM, we typically have a few models that we fit to the data. Just in this case as well, we had a few models that we fit to the data. And here we considered models of different complexity. So for example, a whole 3-level HGF versus a more limited 2-level HGF, where we thought perhaps actually participants don't track the volatility levels. Perhaps the task was too dynamic and there was too much happening in the task for them to actually track it. We also compared it to a more simple model of sort of trial-by-trial learning based on the Rescorla-Wagner model. But the key other feature that we changed in different models was basically if both the responses themselves, but also if the learning rates are specific to a particular context that is relevant. So which means that are there learning rates specific to spatial predictions and specific to temporal predictions? Or are both governed by a single learning rate? And similarly, are both predictions contributing to response equally? And so the model comparison actually revealed that, first of all, we can get rid of the highest level in the model, so we could only have evidence for the lowest 2 levels. But second, and perhaps more importantly, we actually found evidence for specificity of relevant predictions, both in terms of learning rates and in terms of actual contributions to responses. So basically, only the relevant predictions in a given context specifically contributed to responses and also were specifically updated in the relevant context. So what we can do with this model is when we fit it to individual participants' data, we get estimates for predictions and also for prediction errors that participants have in a particular trial. And we can then track them over time and use them as regressors for neural data. So this is indeed what we did here. And we did it in source space. So first we source reconstructed activity evoked by our cues and the targets. And these were located in auditory, visual, and associative regions. And then we extracted time frequency activity from these regions in the alpha, beta, and gamma bands. And we tried to basically correlate these estimates of predictions and prediction errors with the neural activity. So when we looked overall across both relevant and irrelevant contexts, if there were any effects of our predictions or prediction errors on neural activity, we actually did not find any significant clusters. Which was actually surprising to us. But when we looked at specific interactions between predictions and relevance, meaning we were looking at prediction correlates in for example, spatial predictions in the spatial context and temporal predictions in the temporal context, and the same for prediction errors, we found this dissociation between different frequency bands and prediction error signatures. So basically what we found is that relevance predictions were signaled in lower frequency bands in the alpha and beta bands throughout the network, while relevance prediction errors were signaled in higher frequency bands, so in sort of high beta and gamma bands, mostly in the posterior regions where the targets were presented, so in the visual modality. So to conclude this study, we showed that task relevance modulates the influence of predictions on behavior, also in these very dynamic contexts, and that predictions and prediction errors could be linked to dissociable induced neural responses, so in the lower and higher frequency bands. But both of these paradigms that I described until now, they were quite complex, right? So actually participants often did not even know what was happening. They were just pressing buttons, but they would come out of this kind of quite confused. So this actually taught me that perhaps we need to, scale it down a bit and try to find paradigms that can be studied in more detail using more detailed methods. So for example, in passive recordings, but also in invasive recordings. So this brings me to sequence processing. So as we all know, first of all, mismatch responses that I described in the first study, but also these kinds of very complex learning paradigms, they are not the only paradigms where we can study predictions. So sequences are also a very common stimulus domain in which we use both predictions but also some memories. For example, when I talk to someone, I typically can predict what they will say next, at least perhaps the next word, but I also have some memories of what they just said before, right? And these sequences are also in non-speech domains, for example, in music processing, also in non-auditory domains, for example, in gesture communication. But they are also commonly studied in other animals. So, for example, in communication in other primates, other mammals, as well as in birds. So a recent study of my collaborator Lucia Meloni and colleagues used this kind of a sequence processing paradigm to actually identify some slightly different signatures of predictability or prediction that we typically discuss. So these are actually spectral signatures, so signatures in the frequency domain, where this is a study in patients, and when patients were processing these sequences of triplets that repeated over and over again. So for example, Tupiro is such a little triplet that repeats later on in time during a longer sequence of sounds. When they listen to such structured streams of these triplets, their brain activity measured using ECOG showed not only spectral peaks to the syllable level, which was this 4 Hertz peak, but also at the pseudo word, so at triplet level at 1.3 Hertz. And actually there were some regions which showed these peaks almost only to the word rate, to the slow rate, and other regions that showed both word and syllable rate peaks. And if you then randomize the order of these syllables, you don't have these slow peaks anymore. So we took this whole approach as an inspiration to build some studies that we could then use both in humans but also in animals. And this is the work that I've done mostly in Hong Kong. So when we think of these kinds of sequences, we actually have to conclude that it's not just predictive processing, but it's also mnemonic processing. So if I'm processing a particular element of a sequence, for example, the syllable P, I actually have to rely on the memory of the immediately preceding element to know that I'm currently processing a predictable structured triplet, or perhaps this is a condition in which we have a completely random presentation of syllables. I can also rely on the memory of previous segments, so basically the entire sequence, the entire stimulation period, which I've learned, and now I can represent these triplets as some stable chunks or segments. But I can also predict the next element. So basically I can have a content-based prediction of what I will hear next. And finally, we can also think of sequences as governed by temporal predictions. So we can also have these time-based predictions of when I will hear a particular element next. So in this next couple of studies, I will try to answer the the following questions. So first of all, actually, we try to establish a cross-species model of decoding auditory memory, since sequence processing is heavily influenced by memory. But then we want to use this model and test it on sequences with a more predictive component. And finally, we want to disentangle the mechanisms of what and when predictions. So here we focused on auditory sensory memory because first of all, we wanted to test it in humans, but also in rats. So here we wanted to go a bit lower than auditory working memory. So auditory sensory memory is just a low-level subset of auditory working memory, which is thought of as an automatic buffer for acoustic features with no need for active retention. So in the working memory literature, there's been this debate whether working memory is actually accompanied by persistent activity or if it's accompanied by so-called activity silent maintenance. So this activity silent framework has been...
Ti posso chiedere due cose? Ma questo meeting...
I can hear someone. So the activity silent working memory framework has been used to actually develop quite nice stimulation, but also analysis techniques that I will briefly mention now. So the idea here is that following the initial encoding period where we have some stimulus presented to the system, this stimulation, the evoked response might actually differentiate between, for example, stimulus A versus stimulus B. But then during even an activity silent memory retention period, we might actually drop any level of sustained activity and just store the information in some synaptic state. But the key thing here, and the thing that we will also use in the studies, is that you can reactivate the memory by, or at least the activation pattern in the, that differs between the different synaptic states by a so-called neutral impulse stimulus. So if you present now a completely uninformative, for example, noise burst or a very strong visual flash, depending on the state of the network, you will have different outputs. So the same input plus different states corresponds to different outputs. And based on these outputs, you can then decode information about the memory contents. And this has indeed been quite successfully used, even in auditory working memory in humans based on EEG and MEG data. So here we used this principle to actually develop a study that was used in both humans and rodents. So in humans, we had this delayed match to sample paradigm in which participants first heard a very brief tone with a particular frequency, then they heard this neutral impulse during a short retention interval, and finally they heard a probe and they were just pressing a button basically whether the tone, the original tone was higher or lower than the probe. And this was an EEG study in humans. And in a parallel study in rats, we used electrocorticography from the cortical surface of rats covering both primary and secondary auditory cortex. And here we made the paradigm even simpler. So basically we just interleaved these pure tones with noise bursts. And the idea is that we can try to decode the frequency of the preceding tone from the activity evoked by this neutral stimulus, this impulse. So here first we looked at traditional univariate analyses of event-related potentials, and here we found that actually when we look at the tone-evoked activity, so the activity evoked by these tones themselves, we can find parametric effects of the tone frequency already in the univariate analyses. So that means that, for example, the tone evoked amplitude depends on the tone frequency itself. And this was the case both in humans and in rats. And in humans, additionally, we could source localize it to both auditory and frontal regions. But to our surprise, we actually could not find any significant effects of the previous tone frequency on the burst evoked response. So these here, there was no significant effect of the preceding frequency on the tone, on the burst evoked amplitude. However, when we then used multivariate analyses instead of univariate analyses, we could decode this information from burst evoked activity. So we could not only decode the tone frequency from tone evoked activity, which was trivial because we already found these effects in a univariate analysis, we could also recover this information in multivariate analysis, so pooling from multiple channels, multiple EEG or ECOG channels, and using decoding. So I will skip this in the interest of time. But what this shows that again we can decode memories basically just of the immediately preceding tone from a neutral impulse response. So then we applied the same logic to a paradigm in which we actually manipulated predictions as well. So here we had these triplets of sounds. So these were actually artificial vowels, and this is a study only done in rats, again with ECOG recordings. And we repeated these triplets over and over again in predictable sequences, and occasionally we replaced some syllables with these impulse responses. And as a control condition, we had a random sequence where the syllables were reordered randomly. So the key hypothesis here is that since we are dealing with triplets, if we replace any element in a predictable sequence with this burst, we should actually also be able to decode the prediction about what is being replaced by a burst. So for example, if we hear a triplet a o o, and we replace this syllable o, this is what we would predict at the time. So we should be able to decode this prediction in the predictable sequences, but not in the random sequences in which no prediction can be established. So first, I just briefly showed the univariate results, which are basically a replication of the previous study. Here again, we found significant effects of tone-evoked activity, so of tone identity on tone-evoked activity, but again, we did not find any univariate effects of the preceding tone identity on the burst evoked amplitude. However, again, using decoding, we could recover this information. So the key thing here is that we could not only decode the immediately preceding tone, which is again a replication of the previous study, but the key finding is here plotted on the right, where in the predictable blocks shown in blue, we could decode the information about the n-3 tone, which is the tone that is being replaced by the burst. So this is the tone that we would predict to happen, but it's replaced. We can only decode this information in the predictable blocks, but not in the random blocks. And so this difference is also significant between the predictable and the random blocks. So this actually shows that we are tapping into some predictive component here as well. We also run several other analyses on this particular finding. So first of all, we found that actually this decoding increases over time, which shows that we are actually dealing with the gradual learning of this prediction over time. So for example, if we start with a particular triplet, we show that the decoding gradually increases while we stimulate the cortex with the same sequence over and over again. And then when we replace it with another triplet, we actually start from scratch, right? We have to generate new predictions. So again, we drop our decoding and then it increases again during over the course of the new triplet stimulation. So this actually shows that we can tap into some learning of this prediction in the auditory cortex of rats. And finally, I won't have much time to discuss this, but we also looked at whether memory and prediction is actually decoded based on the same features in the data, or is it more likely that they are implemented as in independent populations, for example. So here in a few different analyses based on correlations of topographies, but also on cross-generalization of decoding, we found that actually there was no cross-generalization and no correlation between the spatial arrangement of the features that allow decoding of memory and prediction. So that suggests that we are indeed tapping into different mechanisms, different implementations of memory and prediction, but both of them coexist in the auditory cortex. I think I will skip this study in the interest of time. But I would like to just briefly mention another study that I'm also currently working on. So this is just the last couple of slides. So here, as I mentioned before, when we process sequences, we not only rely on what predictions, so predictions of stimulus contents, but also stimulus timing, so when predictions. And in this study, also based on ECOG, but in humans, we wanted to disentangle the most likely mechanisms of what and when predictions. So in the paradigm, patients here were viewing a series of images and then listening to a tone. So these were audiovisual sequences. And so we had, again, a 2x2 factorial design where the tones could be predictable or unpredictable with respect to its contents and also with respect to its timing. So, for example, in the what predictable blocks, participants had learned some associations between each two images. For example, there was a scene and a face, and they knew that if they saw a White House, they would then see a face of an American president. And similarly, they also had learned some association between each face and the tone. So, for example, if they saw Barack Obama, they knew that they would most likely hear the syllable pa and not ga. But if they saw another president, they might have heard syllable ga and not pa. In the unpredictable conditions, there was no such association between either the images or the images and the tones. The temporal predictability was implemented as a rhythmic manipulation. So here in some blocks, there was a fixed interstimulus interval of 1 second, so they could predict when they will hear a tone. And in other blocks, this was unpredictable, so they could not form such a prediction. So first, looking at ECOG responses, we found actually, again, a dissociation between the two main effects. So we actually found that what predictability mostly modulated activity in frontal regions, while when predictability mostly modulated activity in both premotor and in frontal regions. But we also found an interaction between what and when predictability that influenced activity in sensory regions in the posterior superior temporal gyrus. And again, we use dynamic causal modeling. So coming back to the very first first study that I presented. So this was a similar model, but now additionally, we asked ourselves what kind of, not only connections between regions, but also what kind of gain parameters within regions might actually modulate, might be modulated in both our manipulations of what and when predictions. So here the two candidate mechanisms were a gain parameter that was dependent on inputs from other regions, so a so-called input-dependent gain, which we linked to short-term plasticity, so basically where the sensitivity of a region also depends on what is happening and what is then signaled to this region from other parts of the network. And this is what we found as mediating the what predictability. So here we had an effect on activity-dependent sensory gain modulation. On the other hand, there was another type of gain parameter that we allowed to be modulated by predictability, and this was an input-independent gain parameter. So here, independently of what is happening in the other parts of the network, we could allow the sensitivity of a particular part of the cortex to be modulated by predictability. So this is more akin to classical neuromodulation, for example, dopaminergic or cholinergic neuromodulation. And this effect was linked to temporal predictability. And we actually have then, since that study, also replicated this finding that temporal predictability has a non-specific gain modulatory effect in a couple of different studies. So this showed this interesting dissociation in terms of different gain mechanisms, again, in the auditory cortex. So it seems that all of this is occurring in the auditory cortex, but through different gain mechanisms. And what I'm working on currently, actually in Göttingen, is to look at laminar data recorded from some of the same patients in the same paradigm. So these are laminar probes that span the entire cortical depth, and we can have access to superficial, intermediate, and deep layers more directly, which is quite a unique, actually, opportunity in humans. And what we find in this relatively preliminary analysis is that we can also have some dissociation between the two different kinds of predictability, but also their interaction in the auditory cortex in terms of which layers respond to our manipulations. So here, what predictions actually seem to modulate both superficial and deep layers, while when predictions primarily modulate deep layers. And this is consistent with, for example, optogenetic studies, which shows that deeper layers have more of an influence on slow cortical dynamics in the 1 Hz range and below, whereas superficial layers have a less selective influence on cortical dynamics, so they also influence higher frequency dynamics. So this summarizes the study and the talk. So in this last part, I showed that both memory and prediction can be decoded from auditory cortical activity. And so this actually established paradigms that can be used in all kinds of preparations, in passive listening, also in different animals, and also under anesthesia. We found that memory and prediction are likely encoded by independent populations. And in the study that I skipped, but we also found that if we make these sequences even more complex, we can still decode them from rat auditory cortex, but additional behavioral training might be needed to actually boost more complex sequence decoding. And finally, we found that what and when predictions are mediated by different mechanisms. So what predictions were more linked to short-term plasticity, activity-dependent plasticity, while when predictions were more linked to activity-independent gain control in the auditory cortex. And in this recent opinion piece, we speculate that prediction and memory might similarly be mediated by complementary gain control mechanisms, but likely exerted by another region, for example, by the hippocampus. And actually, in this study that I'm also analyzing now, we have data from the hippocampus. So the idea will be to link these two together. So overall, I showed that attention modulates mismatch responses, especially in these slightly more complex paradigms that are more suitable for human research, but that also we can simplify these paradigms and then actually study predictions more invasively, even in animal models under anesthesia, and their predictions and memories seem to coexist in the auditory cortex, but likely they are implemented by different populations and probably by different mechanisms. So with this, I would like to thank my collaborators, my students in Hong Kong, and mentors, supervisors over the last years. Of course, also to you for your attention. Yeah, thank you, and I'm looking forward to your questions.